I guess it's afternoon now. We have, oh, we're just about to turn over 12 o'clock. There we go. Good afternoon, everyone. And welcome to the Parkinson's Society Southwestern Ontario webinar series for today. We are incredibly lucky to have Morgan Stykel with us today. And before I read her introduction and let her just take it away and blow you away, I do want to do some housekeeping items. So bear with me. As I have mentioned before, we are being recorded. So I hope everyone is aware of that. And the recording will be on the Parkinson's Society Southwestern Ontario website as of probably mid to late next week. You are all muted, but if you have a question for Morgan, which we will have time at the end for a question and answer period, please feel free to put it in either the chat box or the question box, and I'll be monitoring those and bringing them forward for Morgan at the appropriate time. I do want to thank, just before we get to introducing Morgan, a special thank you goes out to our community foundations and United Ways locally for their support of PSSO through the Emergency Community Support Foundation grants. A lot of our local community foundations and United Ways have supported the PSSO education program through recent donations during the pandemic. So I do want to give them a special shout out just to say thank you for supporting groups like us and our work in the community. So let's get started. I've been fortunate to get to know Morgan a little bit over the last couple of days. She's an amazing person, and the research, I think, is just going to blow everyone away. So let's introduce her. Morgan's a PhD student in Dr. Scott Ryan's lab, which I believe is the Molecular and Cellular Biology lab at the University of Guelph.
Yes.
Yep. And where you use Parkinson's patient-derived stem cells to study the disease. With these stem cells, Morgan cultivates disease model systems by recapitulating vulnerable brain populations. Her ongoing research is primarily focused on understanding how alpha synuclein mutations drive increases in reactive oxygen and nitrogen species, which contribute to disease pathology. That's a mouthful. I got to give you credit on that one, Morgan. Notably, the research contributes to the growing body of literature that argues that Parkinson's disease is a multifaceted disease, including or involving redox, stress, and genetics. Her research efforts so far have contributed to over 13 publications and counting in scientific journals that are amongst the most impactful in the field. Her scholarly achievements and involvement in the community have earned her major awards, including the prestigious Vanier Canada Graduate Scholarship. You may have heard us mention, as we were talking prior, she hopes to continue her research and advance her skills at the postdoc level as she works towards her career as an independent researcher. And it sounds like it's going to be an amazing career. So without further ado, bear with us. I'm going to give Morgan control and let her begin her presentation. Morgan, welcome and thank you for joining us today.
Well, thank you so much for inviting me today. Can you see my screen?
We can.
Awesome. Okay. All right, so good afternoon and good morning, depending on where you're listening from today. I'm Morgan Stykel, and I'm a PhD candidate in Dr. Scott Ryan's lab at the University of Guelph in the Department of Molecular and Cellular Biology. And I'd like to start off again by thanking the Parkinson's Society of Southwestern Ontario for this fantastic opportunity to showcase my research in this webinar. And it sounds like one of my neighbors might be hammering, so I hope that isn't interfering too much.
Actually, we're just seeing a blank screen right now, Morgan. Okay. So I think there we go. Perfect. Thank you.
All right. I don't know what I did, but there we go.
That's okay. It's working now.
Right. So I'm going to begin today by giving a little bit of history about myself and what led me to choose research as a career. So I completed my Bachelor of Science at Trent University in Peterborough, Ontario. And during this time, I was highly involved in extracurricular activities. So for example, I was the philosophy departmental representative, and I was the psychology student representative for both third and fourth years. I also helped organize undergraduate conferences, and I even headed my own club that was called WIT, which stood for the Writing Insight Team. In my second year of my Bachelor's of Science, I began volunteering in Dr. Hugo Lehmann's research lab. And I loved it so much that I actually enrolled in 3 separate research practica courses. And then I also did my fourth year honors thesis in his lab. So in his lab, I studied how to make memories resistant to amnesia. And in doing so, I learned numerous surgical techniques. We used rodents and different behavioral testing techniques with those rodents. I learned how to collect different tissues, and I spent a lot of time looking under microscopes at brain samples. And ultimately, this experience fostered my passion for research, and I decided to continue researching and go to graduate school. And I was really, really interested in studying and using stem cells. And so I found a lab that does use stem cells. So I did my master's in neuroscience at the University of Calgary. And here I was part of a really large and growing research group that was headed by Dr. Jeff Berneski. And while his lab is primarily involved in using stem cells for skin regeneration and understanding wound healing, I was part of a smaller group in his lab that uses cells to support peripheral and spinal cord nerve regeneration. So in his lab, I was also mentored by a bunch of postdocs who actually had a really large hand in shaping me as a scientist. They taught me even more research techniques and ultimately allowed me to collaborate in efforts that culminated in 9 publications. And in research, that's our type of currency. And while studying spinal cord and nerve regeneration was fascinating, I really wanted to be studying the brain and neuron biology to tackle complex questions about neurodegenerative diseases. And so I decided to move on and do my PhD in Dr. Scott Ryan's lab at the University of Guelph. And so it's here that I actually use stem cells to study what's happening in Parkinson's disease at a cellular level. So as was mentioned, I have the honor of being a recipient of a Vanier Canada Graduate Scholarship, and my research has contributed to multiple publications which specifically examines Parkinson's disease. And during this talk, I'm going to highlight some of these findings. I've also been a teacher's assistant for cell signaling biology courses, and I ran a journal club that focuses on reviewing papers in the areas of disease, development, and aging. But I'm more than just a scientist. We recently rescued a really sweet dog that we named Wellington. And before COVID, I coached boxing a couple of times a week to various age groups ranging from 5 years old to 60 years old. I also was a member of the competitive fight team. But with the ongoing pandemic, I haven't been to the gym, and I'm choosing research as my primary focus and where I'll be exposed. But I do miss my team, and hopefully, the world will go back to normal in the near future and I'll be able to continue boxing. So I'm going to jump right into it today. And before I begin, I'm going to give an overview of Parkinson's disease. And many people who are tuning in today might be familiar with these details. So I'm just going to just do a brief overview here. So Parkinson's disease is the most common movement disorder, and after Alzheimer's disease is the second most common neurodegenerative disorder. It affects approximately 1% of the population over the age of 65, which means that over 100,000 Canadians are affected by the disease. Nearly 90% of disease diagnosis is idiopathic, which means that there is no known cause, while the remaining 10% of disease diagnosis is inherited through genetics. Parkinson's disease is a progressive disease. I think you can see my mouse at the bottom here. Many patients experience symptoms that are related well before diagnosis occurs. So diagnosis would be on this timeline here at time 0. And diagnosis typically occurs when major motor symptoms start to become evident. And these motor symptoms are classified as Parkinsonisms, and they include slowness of movement, rigidities, and tremors. And these symptoms begin to become evident once the disease has already significantly advanced. So in the healthy brain, dopamine-producing neurons populate the substantia nigra, which is the brain region that hosts those neurons, and there they project to other brain regions to release the dopamine and regulate and attenuate movement. But by the time Parkinson's disease is diagnosed, nearly 70% of these neurons have already degenerated. So many research groups, including ours, are studying why these cells are particularly vulnerable to the disease. And while there's been lots of progress, the answers actually seem to raise a lot more questions. And what's becoming more clear over time is that Parkinson's disease is not just a disease of aging, but it's a combination of genetics and environmental factors that are probably playing a role. So what's happening at a cellular level in Parkinson's disease? In nearly all instances of disease, Parkinson's disease progression correlates with the spread and accumulation of a protein known as alpha-synuclein. And we don't really know what alpha-synuclein is doing in a healthy cell, which makes studying this protein very complicated. But in Parkinson's disease, alpha-synuclein levels increase. Alpha-synuclein misfolds itself into what we call oligomers. These oligomers are believed to be the toxic species, and they obtain many traits that we can use in the lab in order to classify a cell as diseased or not diseased. So some of these traits not only include the increase of the protein, alpha-synuclein, but modifications. So oligomers, or that toxic species that is found in Parkinson's disease, often have modifications such as PS129. PS129 is highly abundant in Parkinson's disease patient brains, while healthy brains or healthy cells, this level is very low. So it's a really good marker of the disease, and I use it throughout my studies. We also know that oligomers are insoluble and they are resistant to degradation. Eventually, these oligomers stack upon themselves and form fibrils. And these fibrils act like a vacuum, sucking up other proteins in the cell and acting as a blockade, resulting in the cell's inability to form its function. It would be like sticking a random garbage dump down the middle of a highway, which blocks traffic, so now people can't get to work and do what they need to do. These blockades are ultimately referred to as Lewy bodies, and they're believed to be the last stage of alpha-synuclein transgression, and they precede neurodegeneration. So in the early 1990s, doctors thought that they could repopulate the Parkinson's disease brain with healthy cells in order to restore function and dopamine levels. So they took midbrains from donated fetuses and grafted those cells into Parkinson's disease patient brains. And you can see that the neurons both survived and integrated. That's what this diagram on the left here is showing. And in the middle is an example of a PET scan, which shows increasing dopamine, increased levels of dopamine. So the red is higher levels of dopamine after grafting. So we have before engraftment, very low levels. 1 year post-engraftment, it starts increasing, and these levels are even more increased at 3 years post-engraftment. Another way to look at whether or not fetal grafts could potentially be a way to restore function and dopamine levels is by looking at the universal Parkinson's disease rating scores. And you can see these triangle graph lines. People that received the fetal grafts, patients that received the fetal grafts had lower scores, meaning they had lower signs of Parkinson's disease, lower symptoms. And while these findings were initially super exciting and promising, the results were contentious. The drugs that people were taking so that they wouldn't reject the fetal grafts seemed to potentially be interfering with the ability to improve patient outcomes. The way the fetal graft dissections were conducted weren't consistent between different labs and different experiments. Some patients may have received fetal grafts that were more enriched for dopaminergic neuron populations than others. Storing the graft cells before transplantation was not consistent between studies, and many cells didn't survive following transplantation. And sometimes the fetal grafts actually caused patients to have dyskinesias due to excessive dopamine. And this is because the excessive cell concentration created hotspots of dopamine in the brain. And of course, there's ethical concerns about using fetal tissue as a therapy. So an alternative cell source is needed. Recently, it was discovered that stem cells can be generated from adult humans. So you can take a small sample of skin cells or blood cells from a person, and you add these specific factors, which ultimately changes the genetics of these cells, causing them to revert back into a stem cell. And these cells go through rigorous testing to make sure they are in fact stem cells. And stem cells are really attractive because, first off, they're an unlimited source of cells. So they replicate themselves indefinitely and you'd never run out. They can form any type of cell in the body, so the research implications are huge. So for example, you take these stem cells and you can generate eye cells, kidney cells, liver cells, or in our case, dopamine producing neurons or brain cells. Then they can be used to study the disease. So you can assess whether different compounds or drugs would be effective in treating the disease. And then those drugs that are effective could potentially be used to treat patients themselves. And in theory, these cells, if there's a genetic mutation that is contributing to the disease or causing the disease, those cells could be repaired using gene targeting methods so that they essentially are now healthy. And then you can generate the cell type of interest that in theory could be transplanted back into the patient with no threat of being rejected. So these are some of the reasons why stem cells might be an attractive alternate cell source and are a very good system to use in a lab. And so this study compares the effectiveness of stem cells to fetal cells. So they model Parkinson's disease here in mouse and monkeys by using a drug to destroy the brain region that receives the dopamine. So they take stem cells and they generate dopamine producing neurons, or they compare that to the grafting of fetal cells into those Parkinson's disease model systems in the monkeys or mice. And then they assessed whether or not that was effective. And so you can see here that the stem cells were able to survive and integrate throughout the brain. And in fact, this graph down here shows that the stem cells were actually a more bona fide dopamine producing neuron. So the higher levels of the red and the green bars here are what we were looking for, or what they were looking for. And you can see the fetal cells have a lot less, they were a lot less authentic and more of a mixed population. And I don't show it here, but the stem cells were also able to rescue some of the movement impairments that the initial modeling of the disease caused in these animals. So in sum, stem cells were just as effective as the fetal cells at repopulating the degenerated neurons. The stem cells were potentially a better option because the culture systems can be controlled and purified so that only the cell type of interest is grafted. And this makes the stem cells a more reproducible and robust system. And of course, there's not as many ethical concerns when we use stem cells that are derived from adult humans as opposed to fetal cells. However, in the last decade, it's become evident that grafted cells might actually be susceptible to disease. And so in the bottom here, the bottom part of the panel, you can see that alpha-synuclein staining shows these large deposits in the substantia nigra where the dopamine producing neurons are, and they show signs of misfolding, which these white dots mark here. And in the grafted cells on the top panel, you can see that those cells also are showing large deposits of alpha-synuclein that's misfolded, suggesting that the grafted cells are also susceptible to disease pathology. And these are signs of Lewy body pathology. So these cells are likely going to die. This led to my primary research question: does alpha-synuclein transmit from cell to cell? And if it does, how does this occur? So in our lab, we use patient-derived stem cells to study Parkinson's disease. So this patient has an A53T mutation, which is known to cause Parkinson's disease at an early stage. These cells have been genetically modified so that that mutation was actually corrected. This gives us a set of identical cells to work with the sole difference being the existence of the A53T mutation or not. So at the end of the day, we end up with identical cells, but one of the sets has Parkinson's disease and one of the sets is a healthy control. And then we generate the cell type of interest that is most susceptible to degeneration in Parkinson's disease, so a dopamine-producing brain cell, or a neuron. And we do that by adding specific factors that pattern what the cells would be exposed to during development. So we start at stem cells, add factors, and at the end, as we mature these cells into neurons or brain cells that produce dopamine, now we can compare and contrast the Parkinson's disease or the A53 T cells to our healthy cells in order to understand the disease better. And on the bottom here, I show part of our quality control checks along the way to make sure that we have, at the end, a pure population of dopamine-producing neurons. So what does Parkinson's disease look like in a dopamine-producing cell? Well, we can look for many types of cellular dysfunctions. These include increases in stress markers, even changes to DNA. Cellular organelles or cellular organs such as the mitochondria have different telltale signs that they're dysfunctioning. Cell trash systems that are supposed to clear unwanted proteins become ineffective, and this actually leads to an aggregation of protein, including the aggregation of alpha-synuclein, which we know is part of a telltale sign of Parkinson's disease. And of course, other systems as well are also modified and can be signs of Parkinson's disease pathology. So in this slide here, I'm showing that our Parkinson's disease neurons have classical markers of Parkinson's disease pathology. So when we look at the healthy cells, these neurons are stained for PS129, which, as you remember from my synuclein slide, was a marker of that oligomeric form of synuclein, or that a marker of disease. So PS129 means bad. It indicates that there is likely disease. In our healthy cells, we have really low levels of that purple stain, so low levels of PS129. When we look at our A53T neurons, or our Parkinson's disease cells, PS129 is highly abundant. There's lots of purple staining there. So ultimately, we quantify these levels, and indeed, there's more PS129 in our A53 T cells than there is in our corrected healthy cells. And we coin that a synuclein pathology, also known as synucleinopathy. So these cells are accumulating synuclein in the form of a pathology that marks disease. And so remember the primary research question I had: does synuclein transfer from cell to cell? So our lab has been at the forefront of studying synuclein transfer. And from the last slide, we know that healthy neurons have really low levels of PS129, and Parkinson's disease neurons have really high levels of PS129. So we decided to take our Parkinson's disease neurons and our healthy neurons and put them in a dish together. So our healthy neurons we infected with this green fluorescing protein, or GFP, so that we could distinguish what was originally healthy and compare it and contrast it to the non-labeled Parkinson's disease cells in a dish. And we see here our green healthy cell is now accumulating levels of PS129, indicating that over time, synuclein is transmitting between the cells, and that what once was a healthy cell and had low levels of PS129 now has very high levels of PS129, and the healthy cells are taking on Parkinson's disease pathology. So here we also show that our Parkinson's disease neurons have mitochondrial dysfunction. So as I mentioned, mitochondria are organs and cells, and they're responsible for producing energy. So there are lots of these in every brain cell, and they need to provide energy to different regions within that cell. And so here I have healthy cells and our Parkinson's disease cells that are labeled with mitochondria DS-red, which is essentially a red fluorescing protein that is sticking to the mitochondria. And you can see that in the healthy cells, they're ribbon-like and very long and connected, but in the Parkinson's disease cells, or the A53 T cells, they are more fragmented. So a cell was considered fragmented if more than 15% of the total mitochondria in that cell were less than 5 microns cubed, and quantified the amount of cells that have fragmented mitochondria. And we show that overall there are more Parkinson's disease cells that have fragmented mitochondria compared to healthy cells. So here you see only about 20% of healthy cells have fragmented mitochondria. Several factors might be causing Parkinson's disease neurons to have fragmented mitochondria, but the most characterized and likely explanation is that the cells are undergoing stress. So fragmented mitochondria are unable to supply energy that the cells require, and it's an early indication of disease. So does co-culture elicit mitochondrial dysfunction in previously healthy cells? So when we co-cultured, again, in the same kind of system, we have our Parkinson's disease neurons mixed with these green healthy cells in a dish. Then we labeled them with that mitochondrial red fluorescing protein, and we measured how many green cells now have fragmented mitochondria. And remember, in the last slide, I showed that there was about 20% of the healthy cells would have fragmented mitochondria. But here you can see that has increased to about 50%. So co-culturing and the spread of synuclein pathology is actually triggering other Parkinson's disease-related pathologies in the previously healthy cells. So another way to see if synuclein pathology can transfer between cells is by using fibrils. So fibrils are essentially synthetic, so they're made in the lab, and they're purified synuclein that has been modified so that they oligomerize, so they are clumped into those tiny little aggregates. And we can inject these into mouse brains. So here I have a picture of an injection site. I hope you can see me outline here where the injection site would be in the mouse brain. It's very small. And we and others have shown that this can elicit Parkinson's disease pathology. And so here's proof of that. So we have a mouse brain here. TH is the region that's these are dopamine producing, dopamine stain here. So where the dopamine producing neurons are releasing dopamine at the site. And then we have PS129, as we know, a marker of Parkinson's disease, and high accumulation that are in regions that are disseminating away from the injection site, suggesting that injection of synuclein alone is enough to cause PS129 pathology in the brain and elicit potentially disease. And others have shown that this also will cause behavioral defects that mimic some symptoms of Parkinson's disease in rodent models. So the next question is, will endogenous levels of synuclein be enough to spread pathology? So we can add concentrated levels of fibrils and show that that can cause the spread of pathology, but what are the amount of synuclein potentially secreted from cells enough to actually cause this spread of pathology throughout the brain? So what we're doing is we are taking our Parkinson's disease neurons and grafting them into mice. So you can see the injection site where all the Parkinson's disease neurons are, and this is labeled in red. And of course, our mouse is not human, but the injected cells are human. So human nuclear antigen, that's what HNA stands for in red here. So we've injected all our Parkinson's disease human cells into the mouse brain. And we're doing ongoing analysis to assess whether these grafts are going to trigger synucleinopathy and even test whether they cause Parkinson's disease-like symptoms in the mice themselves. We can also use our stem cells to generate mini brains. So this is something I started doing in the lab, and I'm looking at these mini brains to look at whether or not synuclein pathology can transmit in a more complex environment, but still in a dish. So here I have a healthy mini brain, and here I have a A53T generated mini brain, so Parkinson's disease brain. In both of them are able to produce lots of brain cells, the green label over here. Both of them have regions that are enriched with dopamine producing neurons, so that's what the TH is there in both the healthy and the Parkinson's disease brain. And the healthy brain has low levels of PS129, while the A53T mini brain has high levels of PS129. So this indicates disease pathology in our A53T mini brain. So I'm going to use this system to study synuclein transmission in the future. So I've been telling you a lot about how our lab is involved in demonstrating that Parkinson's disease can spread between cells. And now I'm going to discuss what we can do about it. And so for the next few slides, I'm going to highlight the findings from our recent publications that I have right here. Okay, So we used an antibody to recognize synucleins. You can think of an antibody that's like a key that will lock a door. And so you need that specific key for the specific protein or that specific door to lock it. Whereas if you had a whole ring of keys that were not specific, you'd never get that door locked. And so you can think of, if we're locking the door is not open, the protein or synuclein can no longer do its function in the cell and it's blocked. So we co-cultured our cells. We have our Parkinson's disease cells and we co-cultured it with our green fluorescing healthy cells. And then we treated it with a non-specific antibody or an anti-synuclein antibody to block the effects of synuclein. And the non-specific antibody wasn't able to rescue PS129. So remember when we co-cultured them before, PS129 levels in that previously healthy cells, so the GFP positive cells, were increased. And this was the case when we used the non-specific antibody. But when we blocked synuclein or closed the door on synuclein, that antibody, the PS129 levels, were actually unable to increase, suggesting that the antibody was effective at blocking the transmission of the disease pathology. We wondered if this would protect from mitochondrial defects that we observed. So like we did before, we co-cultured our Parkinson's disease cells with the healthy cells, A53T and corrected. The corrected were stained with that green, or were infected with that green fluorescent protein, so we could identify what was supposed to be a healthy cell. We treated them with a non-specific antibody or a specific antibody, and we looked at whether or not the mitochondria were fragmented. And so with the non-specific antibody, you see the amount of mitochondria fragmented were about 50%, which is what we observed before. And in the healthy cells alone, remember, it was less than 20%. So yes, the amount of fragmented mitochondria in previously healthy cells is elicited when there is a non-specific block. So Parkinson's disease is triggered. But when you block with a anti-synuclein antibody, that is actually it was effective, and the amount of fragmented mitochondria didn't increase. So we're still just over the 20% mark here, suggesting that it was capable of protecting from the disease. So a number of clinical trials using antibody therapies are ongoing. These trials are currently in phase 1 and phase 2. And there are obvious concerns with this form of treatment because while the antibodies might be able to block the transmission of the disease, it doesn't rescue the cells from the defects that they already had. So the Parkinson's disease cells of the A53T still have highly fragmented mitochondria and still had PS129 pathology. Antibodies have a difficult time of getting into the cells themselves, and they require prolonged and repeated dosing. And one of the chief problems of these therapies is that the function of synuclein isn't known. So we might not actually want to be blocking synuclein's role in its environment. So a lot of research groups are actually looking into developing specific antibodies that recognize that oligomeric or that toxic form of that phosphorylated PS129 type of synuclein. And so that kind of research is ongoing in other labs in the world. You might remember that I mentioned in Parkinson's disease, one of the cellular dysfunctions was changes in gene expression. So we compared the gene expression in our Parkinson's disease neurons to their healthy control cells. We found about 6,000 cells were differentially expressed. So they were either higher or lower, but they were different in our Parkinson's disease neurons than our healthy cells. And we compared this to gene expression from healthy brain samples and Parkinson's disease brain samples, which identified about 7,500 differentially expressed genes. Then we compared these, Venn diagram here, but 2,500 of those genes were overlapping. So our cells had about 2,500 different genes that were expressed differently in Parkinson's disease that also overlapped with gene expression changes that are observed in patients. So we looked at these genes, and we found that when we did pathway analysis, they aligned with an altered antioxidant pathway, and that a central antioxidant pathway gene that is misexpressed is a gene called NRF2. So NRF2 is a gene that's involved in neurite outgrowth. So dopamine-producing neurons need these long stems in order to reach cells that are far away and give dopamine to various regions of the brain. So we looked to see if there was a problem in neurite outgrowth in our Parkinson's disease neurons. So here we have some staining where we can visualize the cytoskeleton with MAP2, so it's microtubule associated protein. It allows us to visualize the cytoskeleton of our neurons or our brain cells. TH is a marker of a dopamine producing neuron. And so you can see that we have stained the cells with that. And so here we're looking at both a dopamine producing neuron culture, and we can trace that using different computer programs. And so when we trace it, then we can do what's called shoal analysis. So ultimately this is laying concentric rings, and then we count the number of intersections from the center of the cell where the genetic material is stored. And we count how many times the neurons or neurites, the little stems that project from the center intersect with these rings. And you can see in this graph here, the healthy cells have more intersections than the Parkinson's disease cells, the little pink, the pink line with the black boxes have lower intersections, which means that the healthy cells are more complex, and that's lacking in a Parkinson's disease cell. We can also measure how far away these extensions extend from the center of the cell, and indeed our healthy cells, the corrected cells, have longer extensions than our Parkinson's disease cells. So that means that there is a neurite pathology in our Parkinson's disease neurons. So we wondered if we activated NRF2, if we could rescue these deficits. So we added DMF, which in the pharmaceutical world is known as Tecfidera, and this is a drug used for the treatment of multiple sclerosis. And DMF is a known activator of NRF2. So remember, before we added DMF, we had really low complexity, these black squares here in our Parkinson's disease neurons. After the addition of DMF, the green bar here, it's similar to our healthy cells, suggesting that it was able to increase the complexity of these neurons. And here I show that it was also able to increase the dendritic length, so how far away the extensions would spread from the center of the cell in our Parkinson's disease neurons. So another study we did looks at a protein known as cardiolipin. So after a lot of research, we discovered that our Parkinson's disease neurons, the cardiolipin relocates from the inside of the mitochondria to the outside of the mitochondria. And we wondered, what's it doing there? So we took purified cardiolipin and synuclein fibrils, and we put them in a test tube together, and we were able to measure the shape of synuclein. And we found that with increasing concentrations of cardiolipin, we were able to refold synuclein into a form that looked like its native form, or the healthy form of synuclein. So in other words, cardiolipin actually pulls synuclein out of oligomers, so we were able to revert it back into the native synuclein form, disassembling the aggregate. So ongoing studies in our lab are involved in making vesicles that are rich in cardiolipin in order to disassemble these oligomer aggregates in our cells. So in order to test whether or not this is effective, we take neurons and we treat them with either saline or preformed fibrils. So this is the synthetic form of oligomers that we add to the cells, and then you can see that this increases Parkinson's disease pathology. So this treatment causes PS129 to increase, so they become Parkinson's disease. And we can see that here in this protein blot, where we have low levels of synuclein, this bar here, in our saline treated group, or PBS control here, vehicle. And then when you add the fibrils, or the oligomeric form of synuclein, we actually increase the amount of total synuclein in our cells, indicative of Parkinson's disease. Then when we add different concentration of vesicles that contain cardiolipin, we can see that we are able to rescue the amount of synuclein in the cell back to what looks like a healthy cell. I'm not sure what my time is.
You're good.
I'm good? Okay, so I only have a few more slides, so I will tell you about this study as well, which is really great. So another finding in our lab is that there exists a gene by environment interaction in Parkinson's disease. So we looked at the effect of agrochemicals in disease. So incidences of Parkinson's disease is higher in agricultural communities and possibly associated with the use of specific chemicals. And these chemicals include paraquat and manab, which is a pesticide and herbicide combination that are often used together. So I use them together in our study here, and rotenone. And so what's important is that during these studies, I use pesticides at levels that are below the EPA reported lowest observable effects. So what we would never be exposed to, lower than what we would be exposed to in the real world. And I demonstrate that healthy cells are unaffected by these levels of pesticides, but our Parkinson's disease cells are vulnerable to these low levels of pesticides. So to look at that, I examined mitochondrial trafficking. So I labeled the cells with that mitochondrial red fluorescent marker that I used before, and then we measured in real time how much movement the mitochondria were doing, either away from the center of the cell, so towards the end of those extensions, or towards the center of the cell. And we found that there was a defect in movement away from the cell in only our Parkinson's disease neurons, so these red bars, after we treated with rotenone or paraquat and manab of mitochondria. So the mitochondria are unable to get to where it needs to be after the addition of the pesticides. So mutant synuclein increases this vulnerability to stressors. Why? Pesticides are known to increase oxidative stress, and one type of this stress is nitric oxide. So here's a picture of neurons. DMSO is a vehicle control, so it is not a pesticide. It does not increase oxidative stress. We have low levels of nitric oxide in these cells. So here it's a heat map. So the redder the pixel, the higher the amount of nitric oxide. So after the addition of rotenone or paraquat and manab, you see this large induction of nitric oxide. And so we measure that here. And then we know that we can block the production of nitric oxide by using this drug called L-name. So when we treat with rotenone and L-name, there is no induction of nitric oxide, or that level is highly blocked anyway. And so that is quantified in this graph here. And then we look to see whether or not that was capable of rescuing it in our mitochondrial trafficking. So we added rotenone to our healthy cells or our Parkinson's disease cells, and we see that rotenone prevents anterograde trafficking or mitochondrial movement away from the center of the cell. But when you added L-name, it rescued it, and it was no longer different than healthy cells. And so we were able to protect from that pathology happening. And the same was the case in our paraquat manab treated cells. L-name was able to rescue it. Well, L-name isn't an effective treatment that can be used in patients because it would lead to changes in cardiovascular activity. This is a potential therapeutic avenue to look at different measures of oxidative stress and blocking them. And it's interesting to note that our disease cells might be highly susceptible to stressors, while our healthy cells are unaffected. So in summary, I have provided an overview of Parkinson's disease at a molecular level. I highlighted the role of alpha-synuclein, specifically the role of its misfolded oligomer form, which stands for PS129, which is a marker of disease. I discussed the studies that led to my current research objectives. So I talked about how transplantation of the cell of fetal grafts became sick and had Parkinson's disease pathology following the graft. I describe how we can study whether or not Parkinson's disease spreads between cells in co-culture systems, if we inject them into mice models, and also how we will use mini brains to look at the transmission in a more complex environment, but in a dish, so without having to use live animals. I show that the transmitted synuclein elicits different Parkinson's disease pathologies. So it transmits protein aggregation in the form of PS129. I showed that the co-culture caused mitochondrial dysfunction in previously healthy cells. I summarize our lab's most recent scientific contributions that included problems in gene expression and oxidative stress. And then I also discussed potential therapies that could be used, including antibody therapy, DMF, L-name, and potentially even cardiolipin vesicles. So in summary, we potentially suspect that a combination a combinatorial therapy might be most effective down the line. Grafting stem cell derived dopamine producing neurons plus the addition of an effective drug and genetically modifying the graft so that they're resistant to the spread of disease might benefit future therapies. So I'd like to thank my advisor, Dr. Scott Ryan, lab manager, Carla, who helps in many of these experiments, past and present lab members. There's quite a few, so I didn't list them all, but everyone is appreciated, our collaborators, and please feel free to email me if you don't get your questions answered during the Q&A period following this, and you can find me on LinkedIn as well. And I'd like to thank my funding sources for making all this research possible. And that's all.
Wonderful. Thank you, Morgan. That was an incredible presentation. We do have a few questions if you're open to answering them.
I think so, yeah.
Okay, so the first one comes from Brian, and it says, if alpha-synuclein protein begins in the gut and works up to the brain, is it possible you might have more success transferring stem cells into the gut and change the protein at an earlier stage of computer flickered there?
Okay, I think you're kind of cutting in and out, but I think I got the majority of that question. Okay. So the idea of transplanting the cells is to repopulate the dopamine producing neurons in the brain. So dopamine is needed in the brain to attenuate movement, and it helps control movement. And so it's important that the fetal cells or the cells of interest are actually regenerating or repopulating that brain region that has degenerated and allowing dopamine to get to those the cells that need it. So if you graft in those cells into the gut, it wouldn't be effective because you're not able to provide dopamine to the cells that need it in the brain. If alpha-synuclein is starting in the gut, potentially we need to be looking at a gut microbiome type therapy that can help halt the spread of the disease. But the transplantation of cells would also just be susceptible to that synuclein transmission in the gut. So I don't know, I hope that kind of answered the question.
Okay. Second question goes back to fairly early in your presentation. And it's you mentioned about genetics and environmental factors as contributing to the PD. Does your research point to Parkinson's disease being passed down genetically through families? And then the follow-up to that is, what environmental factors were you alluding to as well?
All right. So near the end of the talk, I talked about specific environmental factors. So the use of herbicides and pesticides, so paraquat and manab and rotenone as a potential risk factor or risk-causing factor. And yes, Parkinson's disease can be inherited through genetics. So about 10% of patients have a disease, Parkinson's disease, that is caused by genetic mutations. And so that's what I used in my study. The A53T neurons were from a patient who had Parkinson's disease. So those cells were highly susceptible to synuclein misfolding, and that's what makes them an effective tool to use in the lab. But there are increasingly a number of other mutations that are being found in and around where the synuclein gene is located. So if you take all the DNA, the region that synuclein is extending just before it and just after it, there are lots of mutations that are becoming more and more associated with Parkinson's disease, so they're being identified. And that's actually something that I'm hoping to study a little more during my postdoc.
Okay, thank you. The final question that we have so far, unless anybody else puts one in, is you talked about stem cells as a treatment. How far away is that realistically from becoming a treatment?
So right now there are 6 ongoing clinical trials that are using stem cells, so either fetal stem cells or adult-derived stem cells as a therapy. None of those results have been out. They're in phase 1 and phase 2 clinical trials right now, and most of them were expected to end at the end of 2020. So I suspect in the near future, we will start to see how effective those are and the safety of the use of the stem cells. These clinical trials are happening mostly in the UK right now. So yeah, I hope that answered the question. So it is happening. We just need to wait.
It is excellent. Well, that's good to know. A lot of our clients are hearing about things, but they never know how far, or whether they're just in the theoretical stage or they're in the testing stage. So that's wonderful to know.
Yeah, it still has a long ways to go, right? Phase 1 and phase 2 is about safety and efficacy, and it usually there's a very small number of participants that are allowed into those types of clinical trials, and they have it's very stringent to get into those types of trials, right? You can't have any other transplantations or any other medical issues, and when you're talking about an older population, it's harder and harder to find people who have never had any other issues as well. So hopefully, phase 2 and phase 3 trials will start to begin, but those can take still a number of years. So it is still a long ways off of knowing whether or not these trials will be really effective, but they are in progress.
Excellent. We had another question pop up as you were speaking. So this one again from Brian. Have your results been replicated by any other labs?
Yeah, so the mitochondrial dysfunction, that's something that a lot of labs have seen, and the changes in gene expression have been replicated. Noticing that NRF2 is potentially a common target in Parkinson's disease. We're not the only lab who has looked at that, and there are ongoing animal trials that are looking at activating NRF2 in order to rescue different pathologies in Parkinson's disease. So most of these things, yes, have been replicated kind of in their own way, like potentially slightly different ways, but the main theme and the main ideas that are happening are replicated and are known in the community.
Excellent. I believe that's the end of the questions, Morgan. I do want to thank you. That was an incredible presentation.
Well, thank you.
The research you're doing is so fascinating, and it's done. I don't even know if I can fathom the molecular level you're working at. Some of those, working in the gene sequences and things like that. So thank you for your work in the field and for moving Parkinson's disease forward. There's so many people that have it and are looking for that hope. And I think your research is part of the path we're on towards giving everyone hope and an eventual cure or more stringent treatment of the disease that will make them more comfortable and easier, to accept their disease. Sorry, I'm just seeing we've got one more question that just came in. If you're up for it. It says, do you think your work may have positive results on non-motor symptoms?
Yeah, I do, especially because we don't know what the role of synuclein is, but by repopulating the region of cells that have degenerated, hopefully it can do more than just rescue the non-motor symptoms. But as the synuclein pathology spreads throughout the brain, it affects the cortex and causes a lot of non-motor symptoms. So if we can find a compound, a drug to block that spread of synuclein, hopefully that progression will just stop. And yeah, it will do hopefully more than just motor symptoms.
Excellent, thank you. I'm gonna, obviously, thank you again for your time. Do have to do a few PSAs, folks. Thank you for joining us and listening today. Parkinson's Society Southwestern Ontario is your voice. We are your organization for Southwestern Ontario. And just to remind everyone of what's going on, we do have our coffee klatch support groups, vocal programs, exercise programs, and our counseling programs available to all of our clients throughout Southwestern Ontario. So please make use of those programs during the pandemic. We also have something new. You may have received a call from one of our wellness callers, wonderful people that are just calling to make sure you're doing okay. And it's It's a chance for you to let them know if you need some support, if you need a little bit of help, if you just want to talk. And it's okay. We're all in the same situation right now, but we want you to make sure that we're looking out for you. Our next webinar is March 24th at noon. It'll be Dr. Alexandra Peel speaking about Parkinson's in the older adult. Followed the next month on Saturday, April 24th. I think we have something for the 24th, it sounds, with our Living Well conference, which this year's theme is Mind, Body, and Soul. It's Saturday, April 24th, virtually from 9 a.m. until 1 p.m. Finally, again, thank you, Morgan. Enjoyed speaking with you. Enjoyed spending our time together. I love your puppy. Wellington is just adorable. So to everyone, on behalf of Parkinson's Society Southwestern Ontario, stay healthy, wear your masks, wash your hands, and have a wonderful day. Thank you for attending. We'll see you the next time.
Thank you.